Let us continue with our series on ethics in biomedical research. Today we'll be covering a very important topic of ethical considerations in the clinical trials of drugs and other interventions. Clinical trials have been gaining importance in the past few decades. Clinical trial is basically a name given to research which involves prospectively assigning participants to one or more health related intervention. And this intervention could be any of the following: drugs, vaccines, biosimilars, biologics, phytopharmaceuticals, radiopharmaceuticals, diagnostic agents, public health interventions, and so on and so forth. As per the GSR 313E 2016, academic clinical trial is the name given to clinical trials intended for academic purposes in respect of approved drug formulations if they are being proposed for a new indication or a new route of administration or a new dose or a new dosage form. These are known as academic clinical trials. The general ethical guidelines for clinical trials remain the same as the basic tenets of ethics in biomedical research. We have to ensure protection of dignity, rights, safety, and well-being of the participants in a clinical trial. Risk-benefit assessment assumes greater importance in clinical trials. The relevant guidelines and the law of the land have to be followed. This could include the Good Clinical Practices, Declaration of Helsinki, the National Guidelines for Biomedical and Health Research, Drugs and Cosmetic Acts and Rules, etc. At all times, the participants' right to agree for inclusion into a clinical trial or decline continuation in a clinical trial have to be respected by the investigators. It is of utmost importance to maintain privacy and confidentiality of participants in a clinical trial. We have to ensure that at least one member of the research team which is conducting a clinical trial possesses the necessary qualifications and the requisite experience required to conduct this clinical trial appropriately and safely. Approval by ethics committee as per the applicable guidelines is mandatory. In addition, regulatory approvals, if required, should also be sought. All clinical trials should ideally be registered with the clinical trial registry of the country or the region where they are being conducted. This has two implications. One, that individuals having the same disease which is being targeted in the clinical trial have an option to get information about clinical trials, ongoing clinical trials, so that they can register in clinical trials if they want. And secondly, investigators have an opportunity to seek out clinical trials which are similar to the one that they are proposing. Written informed consent assuring quality at every step of the consent procedure has to be ensured. Every clinical trial should include provision of free medical management of adverse events and serious adverse events. All serious adverse events of a clinical trial have to be reported within 24 hours of their occurrence to the sponsor of the trial, to the ethics committee which has approved the trial, and to the regulatory agency. If upon investigation it is found that the serious adverse event was related to the trial, then compensation appropriate to the event has to be offered to the participant. The investigators should offer ancillary care for all non-study related illnesses during the period of trial if possible. If a clinical trial is being conducted in a vulnerable population, the ethics committee has to ensure that there is an appropriate, relevant, and adequate justification for the inclusion of the vulnerable population. Let's look at some of the clinical trials. First and foremost is the clinical drugs or vaccine development, which is the most common type of clinical trial undertaken anywhere. A clinical drug or vaccine trial is divided into 5 phases, starting from phase 0 through phase 4. Phase 0 is basically the exploratory phase, which is usually done in the laboratory with limited human exposure to find out whether the intended target is being modulated by the drug or the vaccine. Promising results in phase 0 trials lead to phase 1 trial where the objective is non-therapeutic, whereby The safety, tolerability, pharmacokinetics or pharmacodynamics of the drug or the vaccine are checked. And phase 1 trial therefore include healthy participants because we are trying to look at the safety of the drug or the vaccine. Phase 2 trials, on the other hand, explore the therapeutic efficacy in case of drugs or immunogenicity in case of vaccines. Hence, participants are selected with very narrow criteria, obviously with the who are suffering from the condition under investigation. And phase 2 trials could also estimate the dose response curve for a clinical drug. Successful phase 2 trials lead on to the phase 3 trials where the therapeutic benefit or protection by vaccines has been demonstrated, and this provides basis for obtaining market approval by the regulatory agencies. After the approval by the regulatory agency, which is the Central Drugs Standards and Controlling Authority, CDSCO in case of India, phase 4 trials or post-approval trials are undertaken, which are important for optimizing the use of the clinical drug or the vaccine that has been approved. So let's look at the ethical consideration in each of these phases of clinical trials of drugs or vaccines. During the phase 1 clinical trial where human participation is started, trial has to be scientifically and ethically sound. Ethics committee and applicable regulatory approvals should be taken. It should be conducted only and only in participants who can give voluntary informed consent themselves. Since there is no direct benefit to the participants, phase 1 trials are being conducted in healthy volunteers to see the side effects. Since this includes healthy volunteers, there should be safeguards to protect in terms of recruitment methods, in terms of payment for participation, there should be no coercion, and the consent procedure should be well documented and appropriate. If phase 1 clinical trial is undertaken in patients with terminal illness, due consideration should be given to the medical condition and the study procedures. Measures to minimize risk while undertaking a phase 1 clinical trial include excluding participants who are at higher risk of side effects of the drug, review the procedures which pose a risk to the participants, evaluate data about the procedure or the drug for serious adverse events, and careful monitoring and early intervention throughout the trial. The study site should have resources and procedures for immediate resuscitation of individuals if required. If a trial is being undertaken with a high-risk intervention product, it should be carried out only in a hospital with experienced personnel and facilities to manage medical emergencies. And phase 1 clinical trials, investigators should try and involve medical pharmacologists or physicians who are trained in clinical pharmacology. For phase 2, 3 and 4 clinical trials, the required ethics committee and applicable regulatory approvals are necessary. Phase 4 clinical trials which would require ethics committee approval would include clinical trials, outcome research, maintenance of registries, and if there is a proposal for a new use or a route or a dosage form of a drug for non-commercial purposes. Phase 4 trial on drugs which have had their market authorization for less than 4 years duration requires regulatory approval. For vaccine studies, the ethics committee approval and considerations remain the same as for drugs, except that for phase 1 vaccine trials, the dose and the route of administration is to be studied to determine safety as well as the desired biological effects. The participants have to be informed that if live attenuated vaccine is being used, there is a small but finite risk of the live attenuated organism producing that specific infection. If clinical trial is to be undertaken with recombinant DNA vaccines, applicable governmental guidelines should be adhered to, and the control group in vaccine trial should receive the complete dose of the effective vaccine after the trial has been completed. They should not be left as such. Now, sometimes bioavailability and bioequivalence studies are undertaken which form a part of a clinical trial. So, what is bioavailability? It is where the proportion of total administered dose reaching the systemic circulation for biological effects is being measured. So we are measuring the proportion of the drug which is available for biological effects. So that is bioavailability. Bioequivalence, as the name suggests, are trials where two or more medicinal products with same active substance are compared for equivalence of their biological effects. Such studies should ideally be scientifically sound. The benefit risk profile of the reference drug or the product and the investigational product should be evaluated very carefully, and careful consideration need to be given to the study procedures for the bioavailability or the bioequivalence study. Such studies are usually conducted in healthy volunteers because of the nature of the study. The ethical considerations in such a study include the recruitment methods, which should be appropriate. If it is to be conducted in a vulnerable population, there should be adequate justification for the same. Then payment for participation should not be such that it coerces the individuals for participation. Consent procedures should be well documented, appropriate. There is a tendency of healthy volunteers to participate in multiple trials, especially with bioavailability and bioequivalence studies, and this should be avoided through maintenance of a volunteer registry. For such studies, the amount of blood that is drawn during the study should be within physiological limits, causing no harm to the participants. Bioavailability and bioequivalence studies sometimes include the use of placebos. So when can placebos be used in a clinical trial? Placebos are used when there is no established effective therapy available that justifies the use of placebo in one of the arms. When withholding the established therapy is not going to expose the participants to serious harm, only then use of a placebo is justified. If the disease itself is a self-limited disease, then placebo can be used for comparison with the drug. And if there is a doubt that using an established therapy as comparator would not yield scientifically reliable results, and at the same time placebo will not be posing a serious risk or harm to the participants, then the use of placebo is justified. However, whenever a placebo is used in a clinical trial, there should be safeguards in place to protect the participants from harm, such as there should be clear withdrawal criteria in case the participant is facing some adverse effects. There should be intensive monitoring, and rescue medications should always be at hand. Usually, studies involving the use of placebo should expose fewer participants to the placebo arm. A 2:1 randomization is most appropriate. An additional arm with an active comparator can sometimes be used as a 2:2:1 ratio, where the first is the investigational product, the second is the active comparator, and the third is a placebo. It should be ensured that the arm which is receiving the placebo can transition to standard of care or the active product after completion of the trial. This brings us to the device trials. Now, what is a device? By definition, medical device is an instrument or apparatus or appliance or implant or any article or software which is intended to be used either for detection, diagnosis, prevention or monitoring of an illness, or for treatment, alleviation of physiological condition or an illness, or for replacement and modification of a congenital deformity, to support or sustain life, or a device which is used to disinfect the medical devices, or for control of conception. So all these are included in the definition of a medical device. So clinical trials involving medical devices should be conducted in accordance with the ICMR Ethical Guidelines 2017, the Indian Good Clinical Practices, as well as the GSR 78E 2017 or as amended from time to time. In such cases, safety data in animals should be obtained first, similar to when a new drug is being used in a clinical trial. So first we should have a safety data in animals so far as possible. The procedure to introduce the device in the patient itself is also to be evaluated for safety apart from the safety of the device itself. The procedure by which the device is being used in the patient should also be evaluated. And always, always, always avoid therapeutic misconception. That is, no patient should be assured that this particular device is going to treat or diagnose the condition with surety. Medical devices are classified into class A, B, C, and D according to the level of risk to the patient. Class A devices which have low risk are the thermometers, bandages, tongue depressors. Class B devices which are the low to moderate risk include hypodermic needles and suction equipment. Class C devices which are moderate to high risk include lung ventilators, bone fixation plates. And high level of risk in class D devices include devices like heart valves and implantable defibrillators and so on. So the device trials for any medical device need to adhere to the guidelines according to the level of risk which is posed. We'll be following up with further ethical considerations in respect to other type of clinical trials in my next presentation. Thank you.